Okay, good afternoon everyone, and welcome to this session of 4 oral communications in the clinical pharmacology section. I should make a declaration at the outset that I'm not a clinical pharmacologist. I've been asked to chair this session at quite short notice, so I would certainly appreciate lots of well-informed questions from the audience rather than me trying to muddle by. But the titles of the talk certainly look interesting. And I'm looking forward to what the speakers have to say. I should probably introduce myself as well. I'm Simon Kennedy from the University of Glasgow. I'm also Honorary Treasurer of the BPS. So our first speaker this afternoon is Robin Ferner, who's from the Institute of Clinical Sciences, University of Birmingham. So welcome, Robin.
Well, thank you very much, and good to be back at the BPS and indeed at UFA. When I was a medical student, we had post-mortem demonstrations at lunchtime, and above the dissecting table was written this motto, Mors ipsa docet succurrere vitae, which I gather means death itself teaches us how to succor life. And when it comes to adverse drug reactions and the use of medicines, this seems to me to be an important lesson. But it's quite difficult to get data on deaths from drugs. There are official statistics, of course, and they come like this. And for those who like big data and numbers rather than stories, they may hold some clues as to what drugs do. There are also rather more full data sets, for example, MHRA's drug analysis prints, but again, very little detail about individual cases, rather more in eutrovigilance databases and WHO. And then, of course, there are published reports in the literature. When it comes to the general public, their awareness of deaths from medicines and from drugs comes from newspaper reports, from broadcasts, and I'm told from social media, this sort of thing. Side effects of drugs kill thousands, which gives you a clue, but not much more. Now, there is, in the case of deaths that are or may be unnatural in England and Wales, a system of investigation which dates back to just after the Norman Conquest and has evolved slightly since then. A judge called a coroner establishes who has died and how, when, and where they died. And inquests are held in public. And some coroners, at least, take their duties seriously enough to call evidence, to assemble witnesses, and to ask experts to give advice. And back in 1994, the Birmingham coroner, Dr. Richard Whittington, and I collected 43 cases from his dusty files in the basement of medicines-related deaths, which we published in the Journal of the Royal Society of Medicine. Those data were difficult to get hold of and made me wheezy because of the dust. But things changed when legislation which had been passed earlier was brought into force in 2013 with regard to what are called Regulation 28 letters on the basis of Regulation 28 of the Coroner's Investigations Regulations 2013. And that said that coroners had a duty to make a report to prevent other deaths if they suspected that their concerns might lead to such prevention. And the PFDs, the letters to prevent future deaths, were to be sent to every interested person who, in the coroner's opinion, should receive it. And the addressees must, that's the law, though there's no way of enforcing it as far as we can see, respond within 56 days. And these documents, the letters, are published sporadically on the www.judiciary.uk website, so it's possible to read them. And whilst it's possible, it's not absolutely straightforward. And my colleague, Georgia Richards in Oxford, has, with Nick DeVito, produced this, the Preventable Death Tracker, which makes publicly available the PFDs in an organized and searchable way, it is said. And that's the URL. When you look at PFDs, they represent only a tiny fraction of all deaths. In 2019, before the COVID epidemic, there were over 530,000 deaths in England and Wales, quite a high proportion of which were reported to coroners because a doctor hadn't seen the person who died within a fortnight or for other reasons, but only 30,000 of which went to inquest, and only 523 about went to coroners who were sufficiently concerned to write PFDs. What was in those PFDs and what of it was related to medicines or drugs? Well, an extremely able pharmacy student, Craig Easton, and my colleague Anthony Cox and I went through 500 PFDs published in the years 2015 to 16 and determined that about 20% of them were related to medicines, most of them, the most frequent being anticoagulants, but then opioids, psychiatric medicines, and non-opioid abuse. With Georgia Richards's web scraper, Harrison France and others went through a much larger set of data and found a similar proportion related to opioids, antidepressants, hypnotics, and anticoagulants, or other therapies and drugs. So here's an example. A man aged 56 has a head injury, has post-traumatic epilepsy. He's treated effectively with lamotrigine and sodium valproate. As an aging man, he gets prostate trouble, we can imagine. He's listed for a cystoscopy under general anesthetic. But somehow, on a urology ward, people don't recognize the importance of his anti-epileptic medicines. They're omitted. He has generalized fits post-operatively. He develops aspiration pneumonia, and he dies. There's an inquest. The coroner expresses concerns, including this one. There was no mechanism in place in the hospital to ensure that essential medicines were prescribed, dispensed, and administered. Well, for those of us who work in hospitals where, for example, patients are nil by mouth before operation, this scenario hardly comes as a surprise, even if it comes as a shock. What was the trust's response? Well, that's not recorded and may never have been submitted to the coroner. We just don't know. Here's another example. A woman aged 74 who is dying from cancer, has pain, and has a fentanyl patch, which are adherent, because if they weren't, there'd be danger from the fentanyl patch lying about. Because it was adherent, at the end of 3 days, she wanted to remove it. A nurse advised her to take a hot bath to soak it off, and she was found dead in the bath with the hot tap running. The coroner determined that the cause was that the heated patch had released excessive fentanyl, and it seems that fentanyl patches are designed to work for at least 3 days and may have, when you take them off, as much fentanyl as they've already released. So they still represent a substantial danger of poisoning. The coroner expressed several concerns. The patient information leaflet warned only of prolonged hot baths. Nurses advised, as I've said, that the patient should remove the spent patch by soaking in the bath, and the GP, when prescribing, had been unclear, and the prescription and the notes were discrepant. Regarding the manufacturer's response, it was, with regard to your request for us to define prolonged and hot, we do not have sufficient data on the pharmacokinetics of fentanyl patches. You may think this did not advance matters. So I hope I've illustrated that PFDs can teach general lessons about medicine safety, but has anyone heard of them? We hope that some in the audience will have heard of them, but they're not generally an important part of people's thoughts about medication safety. So we looked to see how many publications there'd been. We searched Medline and Embase and Web of Science, and we found 83 references, which referred to 46 published papers, of which 16 were full papers, which we retrieved along with 3 others. And of those, 10, in fact, referred specifically to letters to prevent future deaths, of which 8 had been written by one or another of the members of our group. We wondered how much the rest of the world knew about PFDs, and so we searched Nexis, which is a database of newspapers, and looked for terms like prevent and future deaths. The Guardian had several references, the Times, Telegraph had 35, but of those, specific references to these letters relating to medicines in the Guardian 14, in the Times 7, and in the Daily Telegraph only those that had already been reported in other newspapers. This sort of thing, teenager's death after being given antipsychotic, was potentially avoided. This is a man who was given an antipsychotic, olanzapine, became quite ill, recovered after the drug was stopped, and was then re-exposed, developed serious brain damage and died. Some of them are not specifically related to therapeutics. Drug abuse in prison is an important subject. An antipsychotic reaction to doxycycline that causes you to jump out of an aeroplane flying over the Amazon jungle must be relatively uncommon. Alcohol poisoning after university initiation ceremony, less uncommon, but not always fatal, and not really the province of the clinical pharmacology. Likewise, death from cocaine in an Oxford college, and blood clots related to an oral contraceptive pill. Well, the newspapers are interested in the drama, but they're not interested in safety. The coroners, however, are also interested in medicine safety, and their concerns include failures of monitoring during drug treatment, inadequate regulation of drugs, harms from alcohol hand gels, which if you drink them, give you seizures and then you die. Dangers of anticoagulants, which persist even with DOACs. Drugs purchased online, which somehow evade regulatory strictures. Emollients causing fires, and the dangers of tramadol, which was once widely believed to be less dangerous than other opioids, widely but erroneously. So my summary is that PFTs will not be useful unless people know about them, one, and take effective action, two, and we don't at the moment have a measure of effective action because the responses generally are not widely publicized, sometimes never appear, and are difficult to apply generally when they're local responses. So often coroners only express general concerns to local organizations. The addressees' responses, as I've shown, can be insufficient, and the journals and newspapers are not interested. I say that, but there is a website called News on the Web, and my colleague Jeff Aronson in Oxford has looked at the data there. They don't show a huge number of reports, but they do show more than we discovered looking at the 3 so-called quality newspapers. They might prevent more deaths if they were better. If they gave classifications according to ICD or ATC codes, they were machine readable, and they included detailed information on coronial findings. But my conclusions are these: that coroners identify potentially preventable deaths from medicines, that their findings can and could reduce harm from medicines, and their concerns of those and medical examiners worldwide should be widely disseminated, not just to this audience, but to audiences in public. Thank you very much.
Thanks, Robin. That was a really interesting talk. I think we have time for a couple of questions. If anyone wants to ask a question, I think there are 2 microphones approximately halfway down the room.
Hi, thank you. That's a really interesting talk. I'm Miguel Escribano. I'm from the Veterinary Medicines Directorate, so I'm a regulator. I would expect that, you know, these stories from the, well, not the PFDs, sorry, would be captured through the pharmacovigilance systems, through the regulators one day, at least those ones that affect products that are authorized as a medicine. They should be captured, and at least there would be some sort of enforcement or push to the companies to take action if there's something that could be qualified as preventable, I guess.
So, well, there is an example where MHRA has been specifically included in the PFD and where they have taken action, and I briefly referred to it. 2 patients died when they smoked, having emollient-soaked dressings, and set the dressings alight, and they incinerated themselves. And MHRA subsequently reported this, I think, via the Health Safety Investigation Branch, as it was then, or Health Safety Investigation Board, and demonstrated how gauze soaked in white soft paraffin burns furiously whilst ordinary gauze doesn't. And MHRA actually issued a warning. I made a discovery, which is that Google Translate will translate Welsh. This says skin oils, danger. But it doesn't read Welsh. so I can't tell you what it says. So occasionally MHRA is an addressee, and occasionally they take action. But I don't know whether there's anyone here from MHRA who wants to defend their record on dealing with drug safety via PFDs. Well, that speaks for itself, perhaps.
Yeah, I guess. Well, thank you very much.
Any further questions?
Hi there, James Fullerton from the University of Oxford. It's probably more of a philosophical point rather than a question in some ways, but I think your talk speaks to the fact that at the moment all we hear about is a push for big data and a push to try and collate and collect and analyze. And there is something very powerful in storytelling, as you alluded to, capturing these sort of single examples where we can extract significant learning points. In every hospital around this country and around the world, I'd argue, we see similar incidents which are captured by serious incident reports and investigations. And often the learning from those is disseminated perhaps within a department or perhaps within a hospital or at most a region, but rarely nationally and rarely internationally. Can we perceive a solution, as in how, is this a journal which is dedicated to learning such as these? Is this some way that we get a way of categorizing and collating such that people who are interested in big data can actually extract this?
James, this is a very important question, and I'm a bit depressed about the possibility of an important answer. I am a member of the Royal College of Physicians Patient Safety Committee, and we recently heard a lecture from a lady called the Patient Safety Commissioner, who has just been appointed and who may be a focus for this. But because patients themselves now push very hard on matters of safety, I just had the impression that her initial targets were things that there'd been a lot of fuss about, like vaginal mesh, and not things that really will save lives of patients who don't complain. So thank you.
Thanks, Robin, for, of course, always an excellent and interesting presentation. Darren Roberts from Sydney, Australia. We have a database which we can access to look at coronial information. It allows us to look at large numbers of cases. It's an imperfect database. Interestingly, though, we're blocked from reporting cases of incidents that are less than 5 in number because of the potential for identifiability. So that becomes an issue. We can look at large trends, but not smaller cases, such as I think these notable cases that you've shared. I found our indemnity groups tend to be a very useful group for sharing this sort of information. Single case such as this, of which we can see clinically there's important steps we can provide in terms of counselling and guidance to patients. I'm just wondering if that's something that's been explored within the UK system.
If by indemnity groups you mean groups like the Medical Protection Society and the Medical Defence Union, back in 1992 and also in 1991, we went through their files looking at medication errors. But as far as I know, that hasn't been done, and that was published, but as far as I know, that hasn't been done since. And it's a good idea.
I guess what I meant more was that they scan these coronial outputs and then they see something like this and then they send it in their magazines or emails to say, this was a significant case and you should all be aware of it.
That's a very useful point because as far as I know, we haven't engaged with the Medical Defence Union, but MPS may even fund a small study on the PFDs.
Okay, thanks very much, Robin. That was really interesting talk.
Could my colleague from Oxford be about to say something?
If we've got the time. James Fullerton's suggestion about publication, I think, is a good one. Some years ago, I suggested to the BMJ that they should publish accounts of these cases, and they weren't interested. Claire Dyer, who is a legal correspondent and does write for the BMJ from time to time, has published some of these cases, but very few. But I think there is a role for a journal of some sort to be publishing these so that they could be more widely distributed or disseminated.
Thank you.
Thank you. Okay, our next speaker is Caroline Sammer, who's from Geneva University Hospitals. Caroline.
Thank you very much. It's a great honor to be here to present this work on paracetamol-induced hepatotoxicity after therapeutic doses in post-operative patients. So as you're aware, paracetamol is the most widely or one of the most widely used painkiller and antipyretic, but it is also a well-known hepatotoxic agent in overdosing via the formation of the reactive metabolite NAPKI, which forms adducts with hepatic protein. The hepatotoxicity of paracetamol has also been described at therapeutic doses in the presence of certain risk factors, such as CYP induction, which increases NAPKI formation, alcohol intake, and possibly malnutrition and fasting via reduction of glutathione stock, and thus which this prevents detoxification of NAPKI. So we were interested in assessing the link between paracetamol toxicity at therapeutic doses and fasting after surgery. So in this end, we conducted an observational clinical study at Geneva University Hospitals in 100 postoperative patients in orthopedic and visceral surgery on therapeutic doses of paracetamol 4 grams per day. Our main objective was to assess liver toxicity as defined by ALT above 2 times baseline of paracetamol post-surgery in relation to nutritional status. And on an exploratory basis, we wanted to identify novel and early biomarkers of liver toxicity via proteomic and transcriptomic approaches. So we collected plasma samples daily before and after surgery to assess liver test kinetics, albumin and prealbumin. We assessed the number of days fasting, the food intake. We conducted mini nutritional assessment before and after surgery. We assessed communication, comorbidities, measured total dose of paracetamol, the severity and profile of liver toxicity, and the imputability of paracetamol was assessed by a clinical pharmacologist and by a hepatologist. So you see the patient's characteristics here. So the type of surgery, 60% at orthopedic surgery, 40% visceral surgery. The average age of the patient was 66.4, and we have 53% male, 47% females. So overall, and maybe surprisingly, almost 50% of our patients had elevated ALT of more than 2 times baseline value. It was higher in visceral patients, almost 60%, and 40% in orthopedic patients. The magnitude of ALT elevation on average was 2.6. It went up to 29-fold in a patient. The average number of hospital days were 5.5, and apart from the patient who had the 29-fold elevation, we couldn't find any statistically significant link between the length of hospitalization and magnitude of ALT elevation. We then looked at the effect of the number of days fasting on ALT elevation. And as you can see here, the longer the number of days fasting, the higher the ALT elevation. After 2 to 3 days fasting, ALT went up by 2.3 to 2.5, almost 4-fold after 4 days. And for those patients who fasted more than 4 days, the elevation was more than 10-fold. And we could find a direct and significant association between the number of days fasting and the proportion of patients who had ALT elevation above 2 times baseline. We then looked at mean ALT elevation according to albumin value. And again, as you can see, the lower the albumin value, the higher the elevation. The elevation was 4-fold between 25 and 29 grams per liter albumin, and it went up to 12-fold when the albumin was below 24 grams per liter. And here you see the proportion of patients with ALT elevation above twofold baseline value according to albumin and prealbumin values. And we found that all patients with albumin below 24 gram per liter and with a prealbumin below 149 had ALT elevation, and on average that elevation was 12-fold. So really a population at risk of ALT elevation after paracetamol, and probably a population where the dose of paracetamol should be adjusted. So we measured ALT elevation. It's the gold standard in the clinics to follow hepatic tests, but transaminases are not very specific. So on an exploratory basis, we wanted to identify proteomic and transcriptomic markers of paracetamol liver toxicity that could appear earlier than ALT elevation. You might have heard this morning about liquid biopsy and of extracellular vesicles, so we were interested in the microvesicles. Why were we interested with them? Because they're circulating vesicles that are released by most cells. They contain a wide variety of biomolecules, and their composition reflects the content of the cell of origin at the time of their release. As they're rapidly released into the blood after a stimulus, they offer the possibility to be potential early biomarkers. So our hypothesis being that hepatocytes release specific molecules directly into the blood via microvesicles, and thus they would be good candidates for detecting early liver damage. So this is the proteomics discovery phase. We selected the 5 patients and isolated microvesicles at 3 time points. Microvesicles were prepared by differential centrifugation, sedimented, and then the microvesicle enrichment was validated by FACS using CD41 and annexing 5 fluorescence. We used quantitative proteomic strategy with a tandem mass tag with a multiplex mass spectrometry, and we analyzed quantitatively the protein with Proteome Discoverer. And these are the results. We identified 372 proteins and quantified them, and 20 of them were deregulated at least at 1 time point. And out of those 20 proteins, 9 were early modulated, and you see them listed here, those 9 early regulated proteins that could be potential candidates as early biomarkers of paracetamol-induced hepatotoxicity. To better understand mechanism, we conducted a protein aggregation clustering using X-means algorithm, and we could distinguish 5 groups of protein, 5 clusters. Those that were rapidly downregulated in blue and green, cluster 1, 2, and 3. Those that were not there was no variation, and those that were lately downregulated in red, that's cluster 5. And using enrichment, gene ontology enrichment, we were able to identify some biological processes. So for the early cluster 1, 2, and 3, we were able to see that cluster 1 was related to immune response. Cluster 2 and 3 were related to exocytosis, which is interesting to understand the release of microvesicles after hepatotoxic stimulus. And cluster 5 was related to platelet mechanisms and vesicle transport. We then conducted transcriptomics analysis, so looking at circulating microRNA, and our overall aim was to purify and detect plasma microRNA, to see the distribution of those microRNA in plasma fraction, and to conduct, and that's something we want to do later on, a transcriptomic screening of those plasma-derived microvesicles. As a first stage, we were interested in MIR-122, which is a microRNA that's liver-specific. It represents 70% of hepatic MIRs, and it's been shown already that it's a good predictor of ALT elevation in human. The drawback is that it's not specific to paracetamol intoxication. It can be elevated in various hepatic hits. So these are the results for MIR-122. you see that miR-122 was detected in plasma in green, and that there was a steeper increase of miR-122, but not necessarily earlier than AST and ALT. You see that it started peaking at day 3. We then looked at miR-122 in the microvesicles, in the extracellular vesicles, and again, we were able to detect miR-122 in the exosomes, but again, it was quite late at day 5. So in conclusion, this study shows that postoperative visceral and orthopedic patients are particularly at risk of hepatotoxicity from paracetamol. 50% of them developed elevated ALT. The number of fasting days, the value of albumin and prealbumin were all strongly associated with the extent of ALT elevation. So paracetamol maximal dose should be reduced in this population. We showed the value of omics approaches for biomarker discovery in the context of paracetamol-induced hepatotoxicity. With proteomics, we were able to identify 372 protein, 9 were modulated early, linked to immunity and vesicular trafficking. And with transcriptomics, we were able to detect miR-122 in plasma and extracellular vesicles. The outlook is to validate the protein candidates in the entire cohort, and we are planning to analyze the plasma-derived microvesicles in extreme phenotypes, so in real toxicity case of paracetamol in patients taking more than 10 grams. With transcriptomics, we are planning to conduct a broad transcriptomic analysis using an Affymetrix chip with 2,500 microRNAs. So with that, I'd like to thank you for your attention. I thank all the people who participated in this project and all the members of my team, as well as the funding for this project. Thank you very much.
Thank you, Caroline. It's a very interesting talk. Got a question from the audience?
Nick Bateman here from Edinburgh. I'm a proper toxicologist. I don't think an ALT of twice normal is toxic. What I think is that it's an indicator that there may be toxicity happening, and it's very useful in that context. But in this patient group, I've got 2 questions. Firstly, what about weight and dose? You said you'd measured the doses. You didn't say you measured the weights. If you broke down a weight-dose ratio, could you get better, more descriptive analysis? And secondly, did any of these patients actually get proper liver toxicity? In other words, did their INR go up? Did they get an ALT that went up a lot, or they got any evidence of hepatic decompensation?
Thank you for your question. So regarding the second question, once we found out that ALT was going up, paracetamol was stopped. So we didn't have a case
but the damage can be done by that time.
Yeah, so we didn't have a patient who developed liver failure, if that's your question. After like the patient who had a 29-fold liver test elevation, it took some time to go back to normal, but we didn't have a patient with hepatic failure in the cohort.
And did you measure their coagulation?
Yes, we did.
And was it changed?
Yeah, so the INR was not modified.
So that patient therefore did not have liver injury, even though his ALT was serious liver injury, even though his ALT was over whatever it was.
Yeah, so yeah.
Something else. Okay. Yeah. And the age dose thing? Sorry, the dose weight thing, the doses and weights, have you analysed that?
Yeah, we have not analysed yet the dose according to the weight. The patient in our hospital, yeah, they're given 4 grams as routine postoperative pain treatment.
Okay, thanks. We have a question here.
Martin Michel, Mainz, Germany. My questions go in a similar direction, but coming from a different angle. And that is, for most of us, out of the blue, fasting for several times, undergoing surgery, is fortunately not a daily life event. So given that you had ALT elevations which did not really lead to a change in liver function, I wonder whether you have any data how often that may occur in a group of similar conditions but not getting paracetamol?
Yeah, good question. We didn't have a control group without paracetamol, but data has shown that in healthy volunteers, so on the opposite, patients who are not fasting, probably 1/3 of healthy volunteers actually have ALT elevation. So in this particular group of patients who are at particular risk if they get a second hit with another reason to have an hepatic toxicity, they might actually have a clinically relevant hepatic damage.
Okay, and a final question.
Yeah, thank you for this talk, Caroline Sommer. This was very interesting. I have a question regarding the exosomes or the extracellular vesicles. So did you, maybe I missed it, but those were global, right? You didn't do any pull down to make liver-specific exosomes, or?
Yes, we looked specifically at liver-specific microvesicles. Yeah.
Right. Okay.
With FACS, and we specifically tagged the vesicles so that to specifically say that they came from the liver.
Got it. Okay. Maybe sorry, I missed that. So did you find any liver-specific proteins like drug metabolizing enzymes with your proteomics or anything?
No, there was no specific drug metabolizing enzymes. They were the group I showed, but no CYPs, no transporters, no metabolic. Yeah.
Okay. Okay. Thank you.
Thank you.
Okay, thank you very much, Caroline. Our third speaker now. Is Martin Michel from Johannes Gutenberg University.
Yep. Good afternoon, and the words of Monty Python, and now for something completely different, which would mean putting on slides. Oh, here we go. Wonderful. So this is a collaboration, and I should say the analyses I'm presenting today are investigator-initiated. So we had been consulting with the company in the design of this underlying study for their commercial purposes and said, well, if we give you our brains, we want you to collect additional data for our academic interests. And here are the conflicts of interest. So the background of our interest in that is that, at least in many countries, online pharmacies are increasingly used by patients, particularly to obtain non-prescription or over-the-counter medicines. It is, of course, plausible that users of online and classic retail pharmacies differ in their characteristics and thus possibly in their observed therapeutic response. To the best of our knowledge, this has not been studied previously in large cohorts. So we aim to compare customers of online and retail pharmacies obtaining the same products and their therapeutic responses in a large cohort. As I said, this is an investigator-initiated analysis. It's a pre-planned secondary analysis of a published anonymous pharmacy-based patient survey of customers who obtained one of 3 OTC products indicated for the treatment of abdominal pain and cramps. In total, we ended up having more than 1,600 participants in the surveys, with in each group more than 800. And the 3 products under investigation include hyoscine-butyl bromide, a fixed-dose combination of hyoscine-butyl bromide and paracetamol. Thank you for setting the stage in the previous presentation. And peppermint oil, the smallest group. And we performed, based on a pre-specified protocol before any data were observed, the baseline characteristics, including self-reported symptoms and the self-reported symptom relief. And the data are means plus minus SD or percent in a given group. So here's the first part of the baseline characteristic. And that includes one of the most surprising findings to us. All of us felt probably the online customers are younger because they are more internet affine than the retail customers, but the opposite was the case. The patients getting their products from an online pharmacy by average were 13 years older.
and not younger as we had expected. Females were somewhat overrepresented in the online customer group. The most dramatic difference perhaps is repeat users constituted more than 80% of the online customers, whereas in the retail pharmacies there were less than 50%, indicating apparently people for first use, or even of an OTC product, first go to their trusted pharmacy and talk, and then potentially for financial savings go to an online pharmacy to obtain additional supplies if they felt the drug was good for them. Another interesting thing is we asked people, with multiple nominations being possible, what triggered you to choose those products? And physician advice was twice as often a trigger in the online customers compared to the retail pharmacies, whereas pharmacist advice by far was more often in the retail pharmacies. That's not surprising. And triggering from information obtained from the internet and social media was quite similar in both groups and didn't play a big role. We asked the participants to tell us which triggers, in their personal opinion, triggered the complaints, and most frequently named were stress, nutrition, or food intolerance, without any major differences between the groups. And we asked them about the degree of impairment of either work or daily chores, of leisure activities, and sleep disturbances on a 10-point Likert scale, with 10 being the worst possible and 0 being no impairment whatsoever. And again, the two customer groups were more or less the same. Finally, and perhaps most importantly on this slide, we asked them again on a 10-point Likert scale, how bad were your complaints, your stomach cramps, based prior to just taking the drug, and that was about 7 in each group, and I want you to remember that for just a second. When we then, after treatment, asked how did your complaints change, it dropped from about 7 to less than 3, equally in both groups, and looking at those who reported more than at least 50% decrease in complaint intensity, again, the percentage was very similar in both groups. Impairment on work and daily chores, leisure, and sleep, again, reduced a lot, but again, similar for the customers of both sources of the medication. From that, we conclude that users of online and retail pharmacies differ in their baseline characteristics, particularly with regard to age, gender, and first with repeat users. However, they exhibit a comparable self-report therapeutic response, and they are not distinguished by the type, the perceived trigger, or the baseline severity. Of course, these conclusions need to be verified based on independent cohorts using other OTC and perhaps prescription medicines. And I always urge caution in extrapolating such findings, even though 800 plus patients per group seems to be a fairly robust database, at least for these products. Thank you.
Thanks, Martin. We had any comments or questions on this paper? Jeff?
Thanks. There are rogue pharmacies and genuine pharmacies online worldwide. You order something from a rogue pharmacy, pay your money. The best possible outcome is that the tablets don't arrive.
Yes.
Because if they do arrive, they're loose, unidentified, no information, or they may not even be the drug you ordered. We set up an experiment some years ago and ordered rimonabant, for example, online. When the tablets came, they were a benzodiazepine, as I recall.
Yep.
So, mind you, they sent us a free sample of Viagra, so we were quite happy. So my question
I'm
is...
surprised it had such a long-lasting effect on you.
Very welcome. Well, my question is, did you distinguish between these
yes,
two
of
types,
course.
and what are
Yes.
the problems?
Okay, so what we did was recruited the participants via their pharmacies. That is, we selected the pharmacies. And that means for the online pharmacies, those were actually 3 of the big players in the market that all are very well established and clearly are not rogue. But in general terms, your concern is highly appreciated, especially for Viagra-like medicines.
Well, just to say that if you go online to these pharmacies, they have what are supposed to be EU, for example, regulated ascertainment or identification. It looks, some of them copy those mottos, and if you don't click on them,
Right.
you don't get the true information. So if you see one of those, you see the logo, click on it to be taken to the proper websites.
I'm fully in line with you. As I said, in this case, we used big, well-established players like Doc Morris. Yeah. And I think, so far I'm not aware of any information that they provide rogue medication.
Yeah.
Thank you. Very interesting, the difference in age. As you say, completely unexpected. Do you have any explanation why older people are tending to go online? Is it just convenience, do you think?
So we are currently undergoing, so all 3 medications are approved for the use in abdominal cramps and pain, but they all have distinct additional indications. And we have information on which indication someone had as the primary indication, and we are currently analyzing by indication how that works, but that's a much more complex endeavor, and we're not getting the biometric support from the company for that, because basically they said, we give you the data, play with it. And we're happy to have played with that, but that's an important question ongoing. And I have this as actually the only possible thing I can think of is either it's a confounder by indication, or the ladies are more into online shopping. But that's just a personal speculation.
Okay, final question, quickly, please.
Sorry, just a quick one. How fast was the response from the online pharmacies? How quickly would you obtain the drug?
I don't know, but basically we asked the participants to report the improvement at the 2-hour time point.
Okay.
So that was standardized in the questionnaire. How well the patient adhered to it, we don't know, but I have no reason to believe that that would have differed between the online and the retail pharmacy.
The reason I'm asking, I suppose, is whilst people are stating that they have comparable symptom intensity, if you, I suspect you believe there's a difference between, or there may be a difference between online and standard pharmacy because of effectively a psychosocial sort of psychological response, because the pharmacology can't be different between the drugs. Normally, if you were feeling very unwell, you would want the drug immediately, and that would suggest you would go in person to the pharmacy to collect it, to have access to it. If I was going online and I knew it would take a week to arrive, that would suggest there's probably a difference between the groups that you haven't identified as yet.
It's probably largely in the online group chronic customers. We saw 80% were repeat users. And that means it was a chronic or at least repeatedly occurring condition, and they just kept the stack at home.
Thank you.
Okay, yes, final quick question then, please.
Thank you. That's a very interesting topic to work on, because the extent of online users is definitely increasing. So I have 2-3 interrelated questions. One of it is the extent of online use increased enormously during COVID time. So I don't know whether your study was based during the COVID time period, or was it before or after? Sorry, I'll complete the question if you don't mind. Yeah. The second question is, I'm not very clear whether you mentioned that the younger people, younger age group preferred the online, or was it the older group? And you mentioned that the repeat users were more. I just wondered whether you did a multivariate analysis, because repeat users and those, it could be interrelated. So the extent to which you get statistically significant different could be a bit different if you use the multivariate analysis.
Yeah. So please don't nail me on it, but I think the whole data collection period of the study was in 19, so before COVID. It may be my fading memory. But because the primary paper on the overall outcomes already was published last year, so that's one thing. As I said, the online users by average, if anything, were older, by average 13 years. And multivariate analysis is also in the planning, but has not been yet completed.
Thank you.
Okay, thank you very much. Our final speaker this afternoon is Diogo Lopes. Good.
Just waiting for.
Should be up in a second. Okay.
So, good afternoon, everybody. I'm Diogo Lopes. I'm a 4th-year resident of clinical pharmacology, and I work at São João Hospital at Porto in Portugal. And today I'm here to talk about drug prescribing analysis, the burden of orphan drugs in a tertiary hospital budget. First of all, I would like to thank the opportunity to talk about a team that represents a special area of interest. In here, in my first slide, usually I have to explain to my fellow colleagues in Portugal what a clinical pharmacology does, but taking into account the audience, I think I can skip it. But just to give you a preview of what we do in our hospital in the area of medicines, policy and management, we perform quite regularly assessment of quality and trends in hospital drug, and we also try to implement some initiatives to improve investment of drugs, like switching from biological drugs to biosimilar, and provide management and administration to implement these type of policies. So to talk a little bit about orphan drugs and rare diseases, we know that in 1999, the European Commission established that the patients with rare diseases deserve the same quality of treatment as the rest of the population, so they intended to do more and do better. And they came to the conclusion that they need to create incentives for the industry, the pharmaceutical industry, to be more engaged in the participation of creation of new drugs. And so they created incentives like they waived fees to be paid and granted some market exclusivity among some other incentives. And they also stated that to apply it for an orphan drug designation, a drug would have to meet 3 different criteria. As you must know, it would be a life-threatening or chronically debilitating disease that's intended to treat. Second of all, it would be to treat a rare disease or a marketing of a medicine that would not generate sufficient profit to justify the investment. And a third would be that the drug would have not to have other diagnosis or treatment option for that condition. And talk about a little bit about rare disease. We know that rare diseases individually represent a small cluster of individuals, but when we group them all together, we know that in Europe, for example, they are estimated to be around 30 million individuals. They are problematic for society because they reduce life expectancy and represent a burden and loss of quality of life for those patients. They are also associated with high medical costs, not only medical direct costs, but also loss of productivity costs. And development of drugs to treat rare disease has many, many challenges. First of all, in clinical trials, it is difficult to enroll patients. There are small numbers of patients. They are geographically dispersed, and also there is not a total knowledge of how the disease behaves and how its natural history. Other problems are regulatory requirements and ethical concerns, for example, the use of pediatric populations. These development challenges will also translate lately to health technology assessment challenges, because here the governments need to know what is the added value of a drug, and the companies are facing some problems because they have weak clinical trials evidence, right? They have absence of alternative therapy, and they need to take account the social solidarity in the equation of the value of that drug. This will lead to, undoubtedly, to high acquisition costs for our governments. So the case of the Portuguese drug life cycle. We have a national drug regulator that performs an health technology assessment and authorizes the drug to be distributed along the country. Also, we have a specific entity that is a local pharmacy and therapeutics committee that regulates the drug to be used in a particular setting. So here, just to exemplify, there is a prescribing doctor that has a patient with a rare disease. He asks for a local pharmacy and therapeutics committee to give authorization for that drug. The committee evaluates if the drug makes sense in that particular setting, and if the hospital has the budget to dispense that drug, and then it informs our national drug regulator that in turn gives the drug to the patient. This is a multi-step process just to ensure quality, but also to ensure that our system maintains and takes into account some economic considerations. We also must have into account that in Portugal, orphan drug costs are covered by the prescribing hospital. It's not a national budget, but the hospital budget is the money is taken from the hospital specific budget. So what have we done? We retrospectively collected data from our hospital pharmacy from 2021, and we made a list of the top 20 drugs with highest budget impact in our hospital. From that top 20 drugs, we selected the drugs used to treat rare diseases, and then we evaluated parameters such as number of patients treated, cost per patient, and global costs with drugs. We found out that these were the 5 main drugs that were used with highest impact: Zolgensma, galsulfase, eculizumab, nusinersen, and alglucosidase alpha. What we found out that these drugs were used to treat a total of 31 patients, and they represented a total expenditure of 11 million euros just from our hospital. In here you can see this is a total expenditure with the top 20 most expensive that had the highest burden on hospital budget, and you can see that about 1/3 is just to treat rare diseases. In here, you might need glasses. The small blue line is the percentage of patients with rare disease in this top 20 budget. So we have that 0.3% of the patients treated in our hospital represented about 1/3 of all the costs with drugs. Our conclusions: we are not making ethical considerations, but we are just admitting that this type of patients, of course, that need quality drugs, and nowadays they just represent a large proportion of hospital budgets. Where to go, what to do? We need to take into account that drugs that come to treat rare disease nowadays have a lower uncertainty of quality, and we need to create better health and technology assessment methodology that are applicable to this rare disease. We need to improve real-world data to gain an estimation of the real value of these drugs, and we need to exchange information between entities. And one maybe that could be life-changing would be the centralized price negotiation, but that's other topic on itself. So thank you. And questions?
Thank you. Do we have any questions? Maybe I can start off then. I can understand why, if prices are negotiated centrally by the government, you might get a better deal, but so why is it not done that way?
In Portugal, the negotiation is done centrally, but then the price, the acquisition cost, is covered by hospital budget. they negotiate the price for the entire country, but then the budget comes out of the specific hospital that is using the drug.
Oh, I see.
So it's a centralized negotiation, but then the cost will come out of the prescribing hospital.
Okay.
The hospital has some autonomy, right, to make a better deal, but it's largely due to volume. If you treat 30 patients, you'll get a discount, not based on outcomes or anything like that.
Okay. Robin?
Thank you very much. It was a very important presentation because orphan drugs legislation has tilted the table for drug development. Do you have any idea to what extent pharmaceutical companies are now concentrating on orphan drugs and profit rather than, for example, antimicrobials?
I think that the acquisition costs that pharmaceuticals, the drugs are getting pricier. Often drugs are really, really pricey because sometimes we have drugs that are only administered once, like the Zolgensma. So when they tend to negotiate, they don't have an alternative. They promote a drug that is potentially life-changing, so they have put the prices really, really high. And on the other hand, the governments don't have the capability to negotiate because they don't have the real-world evidence. So I think the pharmaceuticals found this as an opportunity because they get discount on the application fees. Sometimes they get waived, and on the other hand, they don't have a government with the capacity to negotiate these discount prices. So when the time has come to gather real-world evidence, the 6 years or 10 years of market exclusive have passed, and they have gained a lot of profits from that drug. So if I had a pharmaceutical company, I would explore this rare disease market vigorously.
And if you were Minister of Health?
Well, we are basically buying the price of hope in here because we don't have, and the media exerts a lot of pressure due to the acquisition, but we can find that some drugs that make a real positive change, right? In the case of cystic fibrosis, we can see a daily impact on patients, but there's a fair amount of uncertainty with other drugs, and it's difficult to say no to a promising drug, but where do you go from there? Proceed to a centralized negotiation and try to lower the price, because I think that would be the most doable action right now, make a centralized negotiation in Europe, because if you have a small country like Portugal, we can't negotiate really the prices, you know.
Thank you.
Okay.
Okay, a question.
Thank you very much, Bridget Mannion from Sydney. We have similar problems in Australia. Do you see a role for health economic analysis or what role do you see for health economic analysis in orphan drug assessments and approvals and institutional support?
Can you repeat the question, please?
Do you see any role for
Yes.
more robust health economic analysis in this situation?
Yeah, what I think is health technology analysis is it's not a recent, but is improving a lot, and it's changing a lot in the past few years. And it's designed majorly, or I think I'm still young, I'm still entering this world, to address problems that are not the same problems with orphan drugs diseases. We need to change the entire methodology to start institutions like cost of illness into the analysis, the spillover costs that sometimes are not put into account in the regular health technology assessments. The other thing, you were talking about what you can do local at the hospital. Was that right?
No, I was just commenting that it's a similar situation in Australia with individual institutions bearing the cost. And because of the cost, you then have loss of equity for other patients.
Of course, that's a real problem, the loss of equity. And like I said, I think we have to improve our health technology assessment methodologies to understand the real added value of these drugs, or just wait until we have real-world data to perform a new health technology assessment. But in that way, we could be depriving patients 2-3 years or more just to get more robust data. And I don't think that would be viable for those type of patients, because we also, in here we have the associations of patients that also have a tremendous power sometimes in the negotiation. And like I said, we are buying the price of hope sometimes, and inequity, it's a real problem in here, especially when we have like our case that 1/3 of the budget goes to 0.3% of the patients. Yeah.
Agreed. Thank you.
Thank you.
Okay, one final very quick question.
Thank you very much. Again, I echo others that this is a very important topic. I just wondered whether who pays for this medicines? Are these patients, is the general population has health insurance, or I mean, how does it work? Who ultimately pays for this cost?
Thank you for the question. We have a national health system, and the price of the patient doesn't have to pay anything. It's all full covered in Portuguese setting.
Thank you.
Yeah.
Okay, thanks very much.
Thank you.
Okay, that's the end of the symposium. I'd like to thank all 4 speakers, and for the audience for your wonderfully well-informed questions. I'm sorry I've eaten into the tea break slightly, but I think there is a few minutes before the next symposia start. Thank you.